Your biology IA can look polished, contain accurate biology, and still lose important marks. Why? Because the investigation is not rewarded for looking long or complicated. It is rewarded for making clear scientific decisions and using evidence to answer one focused question. In this video, I will show you 7 common biology IA mistakes, what each mistake looks like, and what you should do instead. As we go through them, check your own investigation. How many of these mistakes can you identify before submission? Under the current IB biology course, the internal assessment is called the scientific investigation. The task is the same for standard level and higher level students. It contributes only 20% of the final grade, is marked out of the 24, and the report must not exceed 3,000 words. The 24 marks are divided equally between 4 criteria: research design, data analysis, conclusion, and evaluation. That means a beautiful experiment cannot compensate for weak analysis, and a strong graph cannot compensate for an unclear research question. Mistake number 1 is using a research question that is too broad. For example, how does temperature affect enzyme activity? This sounds scientific, but important information is missing. Which enzyme? Which organism? What temperature range? What measurement will represent the reaction rate? A more focused version could be, how does temperature between 10 and 50 degrees Celsius affect the initial rate of hydrogen peroxide breakdown by catalase extracted from potato, measured by the volume of oxygen produced during the first 60 seconds. Now, the independent variable, dependent variable, biological system, and measurement method are clear. Before you begin, ask 4 questions. What exactly am I measuring? Which biological system am I studying? And how will I measure the dependent variable? If these answers are not clear, improve the question before collecting data. Mistake number 2 is simply repeating the classic classroom experiment. Repeating the same question with the same standard procedure does not show enough development. A familiar practical can still be useful, but it should be adapted or extended through a focused new question, a justified variable range, a modified measurement, or a meaningful biological context. Original does not mean unnecessarily complicated. Your investigation still needs to be feasible, safe, ethical, and capable of producing useful data. A simple investigation with a strong justification is better than an impressive idea that cannot be completed properly. Mistake number 3 is collecting data without planning whether the amount will be sufficient. Do not decide the number of values and repeats after the experiment has already started. Plan 4 things in advance: the range of the independent variable, the intervals within that range, the number of repeated measurements, and the precision of the measurement method. Each decision should help you answer the research question. There is no universal magic number of repeats. The appropriate quantity depends on the biological system, natural variation, available time, measurement precision, and the analysis you intend to perform. More data are useful only when they are relevant and reliable. Mistake number 4 is writing a method that another person cannot reproduce. Add some enzyme solution. Keep the temperature constant. Measure the reaction. These instructions are too vague. State exact volumes, concentrations, temperatures, durations, measurement techniques, and control procedure. The reader should be able to reproduce the investigation in principle without guessing what you did. Before finalizing the method, check your materials and quantities, precise procedure, control variables, measurement choices, safety ethics, and environmental impact. Do not add unnecessary instructions, but explain every decision that could affect the evidence. Mistake number 5 is ignoring uncertainty and biological variation. Biological data are rarely perfectly consistent, even when the procedure is carefully controlled. Measurement uncertainty comes from instruments and procedures, such as instrument resolution, reading technique, timing, or the measurement method. Biological variation comes from natural differences among organisms, tissues, and samples. These are not the same, but both may affect your interpretation. Do not delete an anomalous result only because it does not match your prediction. First, investigate whether there is a scientific or methodological reason for it. Any decision to exclude data must be justified clearly. Mistake number 6 is presenting a graph that does not answer the research question. In this example, the question asks about light intensity and photosynthesis, but the graph compares leaf size among plant types. The graph may be correctly drawn, but it is irrelevant to the question. Your analysis should follow a clear pathway: raw data, processing, an appropriate graph or a statistical analysis, biological interpretation, and finally, an answer to the research question. Every step must move the investigation closer to that answer. When analyzing your results, ask, what pattern is present? How large is the variation? What does the calculation show? How certain is the interpretation? And how does this evidence answer the research question? A graph without interpretation is only a picture. A strong conclusion contains 4 connected elements: a direct answer, processed evidence, appropriate consideration of uncertainty, and comparison with accepted scientific knowledge. Do not stop at the hypothesis was correct. Explain what the evidence shows, how strongly it supports the conclusion, and how it relates to biology. Mistake number 7 is writing a generic evaluation. Comments such as good effort, well done, or needs improvement do not identify a scientific weakness. The same problem occurs with phrases such as human error, use more repeats, or use better equipment. Which human error? How could more repeats improve the evidence? Which equipment is better, and what specific problem would it solve? A strong evaluation uses 3 steps. First, identify a specific weakness or limitation. Second, explain its effect on the data or the conclusion. Third, propose a realistic improvement that directly addresses that problem. For example, imagine that leaf discs differed in thickness. Different tissue volumes and diffusion distances could change flotation time independently of the variable being investigated. Use the same cork borer to standardize diameter, choose similar aged leaves from the same leaf region, and measure or standardize thickness separately where practical. That is a matched improvement. Before submission, complete this final check. Is the research question focused? Has the investigation been adapted or extended? Is the quantity of data appropriate? Can the method be reproduced? Have uncertainty and variation been considered? Does the analysis answer the question? And is the evaluation specific and matched to realistic improvement? Remember, more pages do not automatically create a better IA. A better IA comes from better scientific decisions. A focused question, justified methods, relevant data, critical analysis, and a meaningful conclusion. Which of these 7 mistakes was the most useful for your investigation? Write the number in the comments and use the final checklist before you submit. Follow Dr. Reza, bioexam coach, for more IB Biology IA and EE guidance, past paper explanations, and exam strategies.